All right, in the last section, we talked about action potentials, and now we're going to talk about how the action potential really kind of contributes, or how the action potential, I should say, gets from one cell to the next, and that's through a synapse. So when these action potentials reach the ends of the axon, kind of the axon terminus or the axon terminal at the very end there, this is where you basically either have an electrical synapse or a chemical synapse that's going to get that information from one spot to the next. Okay, and the synapse basically is a junction between the two neurons. And if we look at the electrical synapse here, you can see we talked about gap junctions being a way earlier on last year, gap junctions being a way of two cells kind of sharing the cytoplasm, so to speak. So you can see here, you could get ions kind of passing right through these two gap junctions as these things are kind of linked together here. And what these gap junctions basically doing is allowing cell-to-cell communication, in this case, the propagation of an electrical signal. Okay. Now the chemical synapses are the ones that you probably talked about in your psych class, and the one that we went over in class. And basically this is the vast majority of the synapses that you have. And you have a depolarization of a presynaptic neuron. So this would be like where the information is coming from. This is the presynaptic neuron, and then this would be the postsynaptic, or after the synapse. And this is the synaptic cleft. This is where the synapse is actually occurring. And this is what's basically happening is you have neurotransmitters, and that's what we see from these green dots here. Okay, the neurotransmitter is getting released into the synapse, and then those neurotransmitters are interacting with ion channels on this postsynaptic membrane, and you're basically interacting with either the cell body or the dendrite on a postsynaptic type of a neuron. Okay, so the release of these neurotransmitters is going to really elicit some sort of a change. They can be excitatory and kind of cause an increase in the activity in the postsynaptic cell. They can be inhibitory, where they're going to decrease the activity of the postsynaptic cell. Whatever the case may be, you're getting the release of the neurotransmitter from the presynaptic cell and through this synaptic cleft at the synapse there onto the postsynaptic cell. Okay, so this is the one thing that we kind of went over in class. I had you guys talk about this at your tables, and then we went over it really in more detail. In some of that detail, basically what you have is the action potential as it's moving down the membrane, and it doesn't really matter how it's getting there, whether it's saltatory conduction or just kind of a slow moving type of a thing, but it gets from this presynaptic cell. And we talked about the idea that there was three different types of ion channels. You have the stretch gated, voltage gated, and ligand gated. Well, here is an example with this calcium channel of a voltage gated ion channel. So the change in the membrane potential opens up this opens up this calcium channel, and that calcium influx is going to have an effect on the vesicles that are there that are containing the neurotransmitter. Okay, and once these calcium ion kind of comes in, the synaptic vesicles here are going to fuse with the plasma membrane here of this neuron terminal. And you can see that through the process we said of exocytosis, they're going to dump their material into the synaptic cleft. And those neurotransmitters now, we said that these things are made in the cell body. We talked about the whole process of kind of transcription and translation that's going to lead to the production of the protein. That protein is going to be made on the rough ER, and it's going to migrate to the Golgi apparatus. It's going to form vesicles, and then there's motor proteins that are going to move these vesicles along the axons within the neuron, and then eventually those vesicles are going to fuse with the membrane. And when they fuse with the membrane, they dump their neurotransmitter into the cleft. Now these things are going to act as ligands. Okay, so that neurotransmitter is going to act as a ligand, because on this postsynaptic cell now, right here, you've got a bunch of ligand-gated ion channels here, which is what it says right there. Okay, those are the ligand-gated ion channels. So you can see this neurotransmitter here. When it binds to this channel, okay, so this is kind of a blown up version of that. When it binds to that channel, it's going to have an impact, and it's going to allow the sodium and potassium ions to move through the membrane, and it's going to change the membrane potential of this postsynaptic cell. Okay. When it diffuses across and changes that potential, now what you can have is an action potential that gets fired in the postsynaptic cell. And what we had talked about here was just, some of the examples were like when we get scared and our sympathetic nervous system, the accelerans nerve dumps neurotransmitter, dumps the norepinephrine onto the SA node. That's going to increase the permeability because that norepinephrine is going to be the ligand that's going to bind to the sodium potassium channels. And there's some other details in here that we don't need to go through. But essentially what you can think of is they're going to increase the permeability of those ion channels and make it easier to fire an action potential, which is going to result in an increase in the heart rate. And then when we're, our parasympathetic, the rest and digest type of a system, when that returns us back to normal, what that's going to do is you're going to have the vagus nerve dumping the acetylcholine onto the SA node. And what that's going to do is that's going to kind of decrease the excitability, hyperpolarize that membrane, and make the membrane really more closed and less permeable to those ions. And what that's going to act to do is restore the resting membrane potential, or the resting heart rate rather, by lowering that resting membrane potential for a while. Okay, so those are the types of things that we talked about in class. Okay, so if you have any questions, make sure you write them down, and that kind of wraps up the chapter 48 stuff. All right, so we'll talk to you soon.